I don't know. I just selected it, but it's not doing much. Let me see if I'm doing something.
You should slide to the entire screen and then
there. Can you see? Can you see it now?
Yes.
Yeah. Okay.
Good. All right. So let me see how I can do this so I can see my notes too. You guys can still see it?
Yeah.
Okay, good. All right. So I'm just gonna use this here. All right. Hopefully things will work out. Let me know if the next slide doesn't show up. But I really want to thank you, Gabby, for the opportunity to share with all of you what we're doing in the lab. I'm very excited to share for the first time some of my thoughts on an ongoing grant that we just got, and so it'd be great to hear your feedback. And if anyone that wants to join in, I'm more than excited to have more people joining this effort. So just to introduce you a little bit of what I'm going to be talking about today, as Gabriel mentioned in his presentation, I'm fascinated by sound and combining sound, behavior, and phylogenetic tools to understand how signals evolve, communicative signals evolve in toothed whales. And so what I'm going to be doing today is to share the idea behind a project that was just recently funded by NSF Career, which is a grant that kind of provides funding for 5 years, kind of to set up the stage for a particular, a lab and research goals for labs. So, so my career is on the topic of evolution of vocal repertoires and toothed whales, and I'm using the word vocal very loosely. Well, although toothed whales don't produce sounds with their vocal cords, so I'm just gonna use it because it's kind of easier. So what I'm gonna do today is to kind of give you a background of where the ideas behind this grant are based on, most of which is based on my PhD and the work that Gabby has done and some of my other students. Gabby is a collaborator of this grant, as well as other people, and that I'll mention later on. And so what I'm going to do is provide that background, then we'll talk a little bit about the basis to be able to answer the questions for this grant, which will require to generate a collection of the signals produced by multiple toothed whale species in order to estimate the vocal repertoire using number of computational tools. And then how do we transform these characteristics into traits that can be used to test hypotheses using a phylogenetic framework. So that's kind of the whole message of my talk today. And I would like to start, and I don't know if you guys are going to be able to see this, but these are spinner dolphins. I'm going to play a video. Let's see if you guys can hear it and see it. But these are spinner dolphins that I recorded a couple of years ago off the Pacific coast of Costa Rica. And I want to see if you can focus on the sounds that are. So I don't know if you could hear, but there's a bunch of sounds playing in the background, some of which are much more clear than others. The ones that are really clear are those what we call and dolphin and the dolphin world as whistles. These whistles are really tonal sounds that have very clear that can be from which signals can be a number of traits, acoustic traits can be extracted. And these sounds have a number of functions. In some species, particular subsets of whistles can be used for individual identity, but in general can also be used in a number of other contexts. And we don't know much, we do know the context for some of these whistles. We do know a lot about their the structures. We know there's so many studies. If you do a Google search, you're gonna find over 17,000 studies done, you know, or that some, in one way or another, describe the frequency, duration, the context, how they change under different environmental conditions, including anthropogenic noise. So these sounds are the most studied communicative signals in toothed whales. But what we don't really know much about is the evolutionary history of these signals. And as a PhD student, when I moved to Miami to work on my PhD, first thing I did was to do a search about what we knew about these animals. And the first thing that stood out is that we know a lot about their structure, but we don't know a lot about the evolutionary history. And at the time, I found these two really precious papers that were the first ones to propose some of the mechanisms or factors that might have contributed to different aspects of the evolution of these tonal sounds. The first one was by Matthew et al. And they did a review paper where they tested the hypothesis or the contribution of body size on signal frequency. And they found that most of the variation in signal frequency in toothed whales or in cetaceans in general was explained by body size. And the other paper was by Potos et al., which is a gem because it really also played a key in what my research is about. Right? They propose in this paper that whistles are an innovation of dolphins. In other words, that whistles are, you know, unique to these, the lineage of delphinids. And so these were like the only papers at the time that provided some sort of explanation or hypothesis about the evolution of signals, one more associated to one of the most a very important factor, body size, and the other one more related to social structure and sociality. But both of these studies lack a phylogenetic framework, which is super important when we ask evolutionary questions. And so for me to test this hypothesis, the first thing I needed to do was to find a phylogenetic hypothesis with as many species as possible, which was not really available at the time. This is 2003. The most species, the phylogenetic number of species was by Messenger McGuire. And this was a phylogeny that I think had maybe 30 species of cetaceans, and had multiple genes, but they didn't have a very fully resolved phylogeny. So when I asked my committee if I could, you know, work on a phylogeny, they almost had a heart attack because they didn't want to see me around the world trying to collect biopsies and all the logistics and work that entail. So I went into GenBank, which is a very good source of molecular data, and I found out that one of the most common genes that or sequences that are uploaded in GenBank is cytochrome B, which is a very well-known gene to use in phylogenies because it has it's thought to be reliable at lower taxonomic levels due to high substitution rates, and it's good to recover known deeper clades. And so we thought this would be a good gene to use to represent kind of the phylogeny of toothed whales of cetaceans at the time. And we use benchmark clades to test the reliability of the phylogeny based on groups that have been supported independently by other studies, including different types of molecules and morphology. And it is surprising that it has survived. Some, like the phylogenetic hypothesis, for the most part, has been supported ever since new phylogenies have come across with far more data. So I use this phylogeny to test the two hypotheses. And the other thing I needed to do is to think about how do we define a whistle, right? Depending on the authors, whistles can, your interpretation of a whistle can differ. It might be based, particularly if it's present of absence, which can be an arbitrary decision, across species can be defined in terms of the context of the behavior, which in this case, the distribution of the behavior is far more limited than the distribution of the sound themselves and the phylogeny. It also, using these broad concepts, are not ideal because when you think about a whistle, it actually represents a conglomerate of characters like frequency and duration, inflection, and other characters that might have evolved, might vary independently, and therefore have different phylogenetic distributions. So one was having a species-level phylogeny to test this hypothesis, and another one is clarifying what do we mean by whistles, right? These are tonal sounds with a specific, with frequency modulation that are narrow banded with variable duration, and we can dissect these acoustic parameters to test different evolutionary hypotheses. And so when we use this phylogenetic framework and, for example, test the hypothesis that whistles evolve as a unique trait of dolphins, we reject the hypothesis. We actually see that whistles have a much broader distribution than previously thought. And that phylogeny implies that whistles either evolve independently twice, once within siphids, the big whales, and once within the lineage of the pandelphinids, which is a clade that contains the delphinoids and river dolphins and platanista. And that there's a loss of independent loss of whistles within delphinids, including within the clade of phocanids and within dolphins themselves, such as the genus Cephalorhynchus. And the alternative hypothesis is that whistles evolved and the common ancestor of big whales and pandelphinids, and then it was lost subsequently 3 independent times. So these are 2 potential scenarios in which tonal sounds may have evolved, and when I talk about tonal sounds, I'm basically referring to whistles because they're kind of the same thing. So that's in relation to the evolutionary history of whistles, but we also see that whistles that are high in frequency tend to be concentrated within the family delphinids. So what this suggests is that while sociality, a context in which whistles are thought to have evolved, might not be necessarily the co-correlated, evolutionary correlated with whistle evolution, their evolutionary history might be intertwined. And we see that in social dolphins, there's this trend, concentration of tonal sounds that are high in frequency and that are also complex. So in blue, for example, here you see that represents signals that have high number of inflection points are concentrated within delphinids. And we also see that within these animals, these dolphins that have high complexity and their tonal contours, this complexity tends to increase with social group size. So again, while tonal sounds did not necessarily evolve in the context of sociality, their evolutionary history seems to be intertwined with their complexity, with whistle presence. The other thing we learned with the phylogeny is that we were able to test the hypothesis of body size proposed by Matthews. We know the body size is one of the most important morphological factors believed to influence animal signal frequency. Body size and the size of the sound-producing organs correlate, and so the size of the organ can place important physiological constraints on signal production. So, for example, small body sizes that have small sound-producing organs limit animals to produce signals that are relatively high in frequency, which are more subject to attenuation and degradation, and limits the range at which these animals can communicate. And in this paper, what they find is that they found this negative relationship between body size and signal frequency, particularly in maximum, center, and minimum frequency. And they find that the body size explained more than 83% of the variation in signal frequency and cetaceans in general, which is basically everything. But when we take into account the phylogeny, we find not evidence that body size has constrained the evolution maximum frequency. You can see here that when you take into account the phylogeny, like the relationships, right, among species, there's less than 1% of the variation is explained by body size, contrasting to previous work by Matthews, Wan, and Pose. And so this certainly does not imply that such constraints do not exist. No doubt the body size constrains the maximum possible frequencies. But what it does imply is that for maximum frequency, the range of tonal sound frequencies used by cetaceans seems to lie outside the area where morphological and physiological constraints will have an impact. In contrast, we do find evidence and support for that body size has constrained the evolution of tonal sounds minimum frequency. So how low can they go? And this goes along the lines of what other studies have proposed, including Matthews et al. But we do find that size can only explain a portion of the variation. You can see that it varies between 13% to 26%. And this suggests that in the evolutionary history of whales, there has been selection for low-frequency sounds, which enable communication over relatively long distances, and that the degree to which whales have been able to respond to this selection through evolutionary history has been, at least in some cases, constrained by body size. Now, these results have been independently supported by other studies like Martin et al. And also because we have now better technology that allows us to sample the frequency domain of toothed whales in a much broader way, we have learned that whales can produce signals with maximum frequencies that, that are not necessarily related to their body size. So for example, killer whales from Iceland have been reported to produce whistles with maximum frequencies of 75 kilohertz, and these are the largest dolphins, right, in the group. So this is really interesting to me because recently Jensen, a good friend of mine, did a similar analysis with echolocation signals. And we know today better about how toothed whales produce sound. And we know that echolocation signals appear to be produced primarily by the right phony lip pair, which are larger than the left side phony lips. And here he finds that frequency, the body size explained about 50% of the variation in echolocation frequency in all toothed whales. And this further suggests that there is a hyper-allometric relationship between frequency and body size when we talk about echolocation signals. But in contrast, in tonal sounds, which are presumed to be produced primarily by the left phony lips pair, which are smaller, we see that the constraints from body size are significantly reduced in tonal sound frequency. And while so what this suggests is that there's more, they're more released from these constraints, and they're free probably to evolve and more subject to influence from, for example, social structure and environmental factors. But overall, we see a pattern that in both cases, when we talk about minimum frequency and echolocation signal frequency, there appears to be selection for long-range biosonars and communicative signals. But at the same time, I think that differences in size and their sound producing organ can also suggest that there is different selection forces that are happening and that are shaping echolocation and communicative signals in different ways. So they seem to have different evolutionary histories. So just in conclusion for this first part, just to kind of set the ground for what we're doing now in the lab, is that we find that tonal sounds are homologous across toothed whales. There's evidence that they share the same sound-producing mechanisms in organs, so they're homologous. There's also evidence that while sociality is not the main factor shaping the evolution of tonal sounds and toothed whales in general, that their evolutionary history is intertwined with sociality. And we also find evidence that body size has constrained the evolution of tonal sound minimum frequency, but not maximum frequency. But only certain amount of that variation appears to be explained by body size. So these results led me later to think about two two things, which are two different projects or kind of intertwined projects that we're working on. One is that the size can only explain some of the variation in tonal sounds. Tonal sound frequencies, it suggests that there may be some independence in which these signals have been changing over evolutionary time. And that tonal sound complexity, that relationship that we find with social lineages within dolphins, made me think about vocal repertoires, because so far we have primarily studied the acoustic structure of the signals and the context, but we haven't really learned about the collection of the signals that these animals are capable of producing. And we know a lot about that in birds and non-primates. Vocal repertoires at evolution is tied to the environment, but also to social structures. And we'll get to that in a moment. So let me just kind of tell you a little bit about what we're doing with evolutionary changes in both vocal and body size and signal frequency. This is an ongoing analysis, so we haven't published it yet. So we're still working and redoing some of the analysis. But some of the results are really interesting. So these two circles show the evolutionary analysis or evolutionary shifts and body size and signal frequency. And on this side, you have body mass. Oh, sorry. And on the other side, you have minimum frequency, I think it is. And the colors represent how fast that evolutionary rate shift is occurring, if it's accelerating or decelerating. And the other thing I want you to look at are the red dots. So the red dots are the points in the phylogeny, the clades in which an evolutionary rate shift has occurred. So if you look at the one that says body mass, there's an evolutionary rate shift in body mass and delphinids and dolphins, and the clade of dolphins. And if you look at the frequency, you see a red dot and the clade of Delphinoidea, which are the dolphins, the monodontids, and the phocenids, that there's an evolutionary rate shift in frequency. Now, notice that these changes are not happening in the same clade, which suggests that the changes, the evolutionary rate shift and body size are not co-occurring with changes, the rate shifts and frequency. And this suggests independent evolutionary histories. Also notice rate shifts on the evolution of minimum frequency is happening before the changes in body size and rate shifts of body size. And this has been proposed actually by other studies, which are very, very interesting. What this also suggests is that allows us to predict that in whales, a signal that is free to evolve could also contribute to speciation. And our results support this prediction, and that frequency explains at least 68% of the variation in speciation rate. And this is quite interesting because we do know that signal frequencies or acoustic signals have contributed to the speciation of other animal lineages and taxa, including terrestrial mammals, amphibians, and insects, and other arthropods. Acoustic signals play a key role in species recognition, social structure, and reproduction, and thus has been recognized to play a key role in diversification. And this could be also a role that might be happening within the toothed whales. Now, this is preliminary data, and we're still working on it. So we're still redoing some of the analysis. Some of these patterns might change over time. But I think it's very useful to think also and these traits in terms of evolutionary changes. And if these changes are happening, co-occurring, or are happening independently. And that might be explaining why so little of the variation and signal minimum frequency in tonal sounds is explained by body size. Now, the rest of the talk, I'll spend it talking about tonal sound repertoires, because the complexity that we see in our previous studies suggests that there's a lot of variation in the contour of tooth whales, which, as had been shown before, is can be important and can be associated with social structure. So when I talk about vocal repertoires, I'm really talking about the collection of all possible sounds an animal can produce, and it has been shown, as I said before, to be correlated with some aspects or some features of sociality. Just to give you a background of the potential of the vocal repertoire of toothed whales, I don't know, you'll probably be familiar with a recent study that was published in 2023 about the mechanisms by how toothed whales produce sound. We have learned a lot about that. Toothed whales have these air sacs and nasal passages that have these structures called phony lips through which sound production occurs. But this study shows that the whales, just like humans and ravens, are capable of producing sound via 3 different registers. So you can see here, for example, one of the registers is vocal fry, also known as M0 register, and this is when the phony lips are relatively relaxed and produce the collocation signals, and on the right side, the phony lips. And humans, these register sounds like a creaky voice. I always think of Darth Vader when I'm thinking about this type of register. Then the bird sounds appear to be produced with M1 or chest register, which is when these phony lips are slightly stretched. And so you have this rapid production of clicks that are, that we refer as burst. And in our case, in humans, that will sound like our normal speaking voice. And then we have the whistles, which are the product of M2, which is the falsetto, and humans what we call the singing voice, which results in this kind of narrow band and a little bit higher frequencies. So these animals are capable of producing these sounds, and because they have 2 sets of phony lips, they can produce sounds in combination or independently from each side, which results in a really rich collection of features, acoustic features associated to the signal. So you can have, for example, biphonation, which is the production of 2 independent sounds. At the same time, you can have deterministic count, which is like this kind of noisy, going from very tonal to relatively noisy and then back to tonal. You can have sidebands and subharmonics. All of these acoustic features just add more to the richness of their vocal repertoire of toothed whales. So if we combine this ability to produce this really rich set of signals plus their ability to learn, we can basically say that this is perhaps potentially one of the most complex vocal repertoires in the animal kingdom. However, despite of how much we know about these animals and their vocal signals, we still don't have a comprehensive and standardized characterization of their vocal repertoire. And here's where the phylogenetic aspect comes in play, because with phylogeny, we can test a number of hypotheses about the evolution of these vocal repertoires. And we can also test hypotheses about ecological drivers of complexity in these animals. Now, only a tiny portion, to be fair, only a tiny portion of vocal repertoires have been characterized, with those of songbirds and non-primates among the best known. And in these 2 systems, a growing number of phylogenetic comparative studies have shown support for an evolutionary increase in vocal repertoire size with social complexity. However, a lot of these studies rely on literature, data that has been mined from the literature, which doesn't really take away, but it's just show the complexity of having phylogenetic studies that incorporate a number of species in the analysis. One of the things that I think about is that if we want to test the relationship between vocal repertoire size and complexity with social structure, the best lineage to test such hypotheses are the tooth whales. And the reason for that is because tooth whales have a more elaborate vocal repertoire than non-primates, and they have a wider range of social structures than songbirds. And songbirds, song is in general more driven by individual elaboration and mating success, while in tooth whales, acoustic behavior is driven by various social contexts and the strength of the bonding among group members. So we want to test this hypothesis. I believe that tooth whales is the best lineage to do so. And so this is the goal of my lab for the next 5 years, to get a number of species that represent different, that are distributed in the phylogeny, representing different social structures, that we can generate a vocal repertoire and to test hypotheses about the evolution of this of their vocal repertoire in relation to the environment, sociality. And so overall, the goals of this project are going to involve, first of all, we need to find create a protocol and computational flow that allows us to characterize the vocal repertoire in a standardized and comprehensive way across different species within the phylogeny, and that we can then transform this characterization into traits that can then use under a phylogenetic framework to test a number of hypotheses, and also combine it with ecological tools that also allows us to get a better understanding of how the environment can also contribute to the evolution of vocal repertoires. So the first step, which is the characterization of the vocal repertoire, this is where the challenge really is. How do we measure the vocal repertoire of a species that has all these incredible, this ability to produce such a variety of signals, and that is changing over time because they're open learners. And so to really get a good understanding, we need to get species across the phylogeny. Each of these species, we need to have data that comes from different parts of their distribution, so we account for that within species variation. And then from that, we also need to figure out what are the best metrics of vocal repertoire, size and characterization of vocal repertoire's composition and diversity. There are different methods that are available, each of them with their pros and cons. So we need to identify what is the best approach so that we can then convert them into traits for a phylogenetic comparative analysis. Now, this is my, I call it my Christmas list. These are all the species I would like in the analysis. On the right side, you see the phylogeny with the red arrows kind of representing the location of these species, trying to kind of get as many species across the phylogeny, but also species that vary in their social structure as much as possible. And these are all species that are known to produce tonal sounds. Now, I'm starting with tonal sounds because it's what we know, right? So we know that there's a lot of studies. We know what are the species that produce these sounds. So before we get into the entire repertoire, I think it's important to start what we know, because it would allow us to create and identify different tools that can be used for a much more comprehensive analysis. And so what we're working right now is to create a project in GHub where we're going to be uploading all the contours that are extracted for tonal sounds. And we're going to have computational tools. We're going to have protocols and guidelines for all the collaborators that want to contribute to this project. So this is a list of the current collaborators to this work, and most of them are working on similar questions of vocal repertoire, looking at, for example, testing different methods, not just in calculating repertoire size, but also extraction, contour extraction, characterization using machine learning and AI tools. So we're in the process of identifying what are the best computational approaches. In the meantime, I think we're going to be, for the tonal sounds, we're going to be focusing on our work, which is Gabby is familiar, many people are familiar actually with this tool. This is a machine learning tool that combines dynamic time warping to measure signal contour similarity in the frequency domain, and has an adaptive resonant theory neural network, which basically what it does is that allows the network to learn as more signals come into the system and revisits them each category as it reclassifies or classify the new ones that are coming in. And so this is a very well-used and a pretty good approach that can be used with any signal that has a contour. And so what we're doing right now is we're testing different vigilance values. This is the vigilance values is the amount of similarity that you set to compare each of the contours that enter the network, and that decides if it goes into category or not. So usually people uses a 96% of similarity, particularly when they're working within same species. But when we're comparing multiple species, we're going to need to test different vigilance values. So we're testing different ones. So for example, here's an example with the killer whale, where we're using instead a 92%. So any contour that had the shared 92% of their contour are placed in the same category. So we're testing that, and the first kind of result of that is by my PhD student Maya, who has been looking at just using a small sample size, looking at comparing the kind of going over how much sample do we need for each of these animals to kind of get a better, really good understanding or representation of their vocal repertoire. And this particular table, what I'm showing you is a subsample that was of the data that was used to compare across different methods that are used to estimate vocal repertoire size. There are many, and there's the enumeration, capture, recapture, Q2 models, and curve feeding models using, like, for example, Chow Hill numbers, which is our favorite in our lab. But it's important to kind of have an understanding of the variation across these methods, so that we can start thinking about how do we translate these measures into phylogenetic traits. So one trend that I think is interesting here is that regardless of the method that you use to estimate vocal repertoire, bottlenose dolphins always come at the top of vocal repertoire size compared to other species of delphinids, and false killer whales generally fall at the end. So this is one aspect that we're working on, and we're just wrapping up a review paper where we go over all these methods and provide the scripts and protocols that hopefully will become available by the end of the year. We're also thinking about comparing the composition, because vocal repertoire is one number, right? But there's also the composition of the signal. So this is a very rough composition using the contours that we generate with our work. And you can see that if we use a traditional classification based on ascending, concave, constant, those categories that we generally use in a broad way to look at signal contours categories, we can see that there is differences across different species. So for example, the bottlenose dolphin tend to have repertoires that are primarily dominated by sine whistles, which are more complex, whereas, for example, Guyana dolphin, most of the composition of the vocal repertoire is represented by upsweeps. So understanding the composition in addition to just vocal repertoire size is also very important. How am I doing with time, Gabby?
I think you're fine because we're late. So you have like 20 minutes.
Okay.
More to make it an hour if you want.
So so these are things that we're thinking about, but we're also working in different projects that can allow us to kind of get a sense of the variation at different scales. Like within species, between closely related species, within species, thinking in terms of ecotypes or subspecies. So Gabby, I hope it's okay I mentioned the paper that we're working on. This was this is part of Gabby's PhD and then also postdoc with Vincent Janik that we have been working for a number of years because it's a challenging paper. But what this paper has allowed us is to really take a species like Sotalias, Guianensis and Fluviatilis, which have a restricted distribution, and actually been able to have samples of whistles across their distribution to understand the variation of the vocal repertoire. So in this project, which I'm not going to say much, but because we're still in process, we're working on the reviews. What it shows is that while there are some differences between the Sotalia, the riverine, and the coastal, in general, there are really no complete separation in vocal repertoire size between species. It helps us to use, for example, in this case, vocal repertoire size is calculated using the Chow's Hill number, and here represented by the inverse of the Simpson concentration index, which describes the expected number of dominant species in an assemblage, and allows us to see how different populations or places where recordings were made vary in their vocal repertoire. We also use it to kind of understand the effect of group size. There seems to be a small effect here. It still needs to be explored in the future. But it gives us this kind of studies kind of gives us a good understanding of how perhaps local differences in the environment, the level of isolation among populations can influence the vocal repertoire and the composition of each population vocal repertoire. So more to come on that front. We also have been able to look at vocal repertoires within species. In this case, we work with the pantropical spotted dolphins and the eastern tropical Pacific, comparing the coastal subspecies versus the offshore subspecies. And it's very interesting because we found that their vocal repertoires, whether you use richness or, again, the Simpson concentration index, their vocal repertoire size is basically the same in the two subspecies. The differences are primarily in the composition of their vocal repertoires, with the offshore subspecies having a higher proportion of sign whistles and the coastal having more upsweeps, which might reflect differences in group size and social structure, or even the conditions and the environment that might shape the acoustic structure, the contour used by these animals in different environments. We have also talked about, look at the context of within species. So we have been studying bottlenose dolphins, and in Panama, it's an archipelago called Bocas del Toro for a very long time, where we have passive acoustic monitoring regularly. And we had recorders during the COVID pandemic lockdowns, and we were able to kind of get an understanding what happens when either the boats are presence is minimized, or when there's a shift in the boats that are allowed to be in the space, right? In this particular archipelago, most of the boats that you'll find are tour-based boats, and because that collapsed during the pandemic, the only boats that were allowed to be out on the water were those that were transporting people between the islands to get food or to go to the hospital. So there was a decrease in the number of boats, but also the kind of activity at which dolphins were exposed to. And that seems to be translated in the noise levels that we observe, although there is some more variation, possibly because now the sound traps are also recording other sounds that would normally be masked by boats, the high density of boats. And what we find here is that during the lockdown, the diversity of repertoire size, dolphin whistle repertoire size was higher than during normal times, pre-lockdown. And that could be again, for many things. Now the sound trap, there's less noise from both, so it's capturing more signals. But I think it was actually more dolphins now using that space without the boats present, because we had, when we look per signals per hour, we or along the day, we have more dolphins present, or more hours with dolphin signals present in the recordings. So this also kind of gives us a sense of the variation, how repertoire size can vary, you know, based on location, based on local environmental factors, and based on, you know, context. So the studies are just helping us to set up the methodology for the larger analysis, which will require a number of species, a greater number of species from different regions, so that we can have something that is standardized and comparable, and that we can account for some of the potential challenges and biases in measuring repertoire size and composition at this large scale. But the other part of the question is the social aspects. How do we measure sociality at such a large scale? So I think what we plan to do is, for the large phylogenetic analysis, we'll still have to rely on what has been published in the literature, and from that derive characters that represent different social features. I have done that before with in my BMC paper, where we created a really large database, and for each species we kind of try to dissect sociality, for example, based on group stability or group composition, group size, and other aspects. So we just have to update and amplify this database. But also we can select species from different social structures and dive more deeply in the field by using drones and recording systems that can allow us to get a better understanding of individual vocal repertoires and their contribution to the overall repertoire, and how that might change with behavior and social in the different social structures. So that's something that we're working on. Now we have data from bottlenose dolphins, and we're working with Sotalia guianensis in Costa Rica, and hopefully we'll be able to go to Brazil, work with Gabi and Renata and others, with other species, more Sotalia and Inia, to collect a little bit more of the social behavior in relation to their vocal repertoire. And my goal is that ultimately we're going to end up with a lot of data that can be different kinds of matrices based on their vocal repertoire. We could look at the contour repertoire traits as categorical traits. We can use the metrics from the vocal repertoire size that can be, we can look at the consensus across different methods and generate perhaps an index that can be used to score vocal repertoire size. We can use a number of other traits, acoustic traits, that will be anyways extracted from the entire process that we go through. And eventually what I'm hoping is that we'll be able to build these matrices that will then facilitate a number of phylogenetic analysis, such as evolutionary rate shifts, analysis of correlation between different features, social features, and vocal repertoire features, as well as estimating just simply the evolutionary history and distribution of these signals. So in conclusion, summary, I think quantifying the vocal repertoire of open learner species is undoubtedly challenging. There are many things that we still need to think about how that go in the process of characterization. And this is definitely our first step, and really understanding how these animals organize and the evolutionary basis of their communication. So far, what we have are few studies that we have been working together show that there are no major differences in vocal repertoire size in closely related species or within species, but there are differences in the composition, which can be a reflection of their the environment and social structure, and that will require further analysis to look into the respective potential associations between these features. And I think what we could gather from this, and at the end of this study, I hope that using a collection of vocal repertoires will be that would be made available to the public through this GitHub. The goal is to have these open access tools that will contribute to a much more democratized and also more open sharing of data that can also be used by other scientists, that can continue the examination of these hypotheses using this phylogenetic framework that we hope to set up across these 5 years, but also that can continue help us to think about in phylogenetic and evolutionary terms when we talk about these animals' communication, and get us to be better at defining sounds, social behaviors that can then later be compared, right, across the phylogeny and can be used to test hypotheses. And then also in the context of climate change, we know climate change is changing sound propagation in the oceans. We can we think about what are the value of some of these signals, how they might be in the life of these animals, how these changes in sound propagation might select for certain signals over others. Will some of those signals that are more complex and more modulated be lost in these much more noisier environments, and then selection for simpler signals, which could translate in a reduction of communication, right, impact their overall, the way that these animals relate and socialize with one another. So these are things that, I think this project will help us to think and test even more interesting hypotheses in this context. So I want to thank all my students that have contributed one or the other, all the collaborators to this project that have contributed with great ideas, and the months ahead will be important source of feedback for the methods that we need to set up. And if you have any questions, I thank you, Paulo Castro, for this amazing video, and Gabby. So if you have any questions, please let me know.
Hello. Thanks for the talk, Laura. It was amazing.
Let me stop sharing so I can see you.
Yeah. Yeah, that's a...
Não tem como.
Pode perguntar aí, e aí qualquer coisa eu traduzo. Mas... Israel, pode falar então, já que o pessoal lá na UERJ ainda está com problemas técnicos, eu não sei o que está acontecendo.
So, thank you again for your lecture. It was amazing. It's always a pleasure to see your data. I have actually one question. It's mostly technical, but sorry for that. So my question, you show some evolutionary approach, how the signal whistles can change over body size, evolutionary, but we can mention there's any significant difference when you're looking for a species, for example. Did you find something like that? Different individuals with different water size in the same species that are different frequencies. Okay.
Yeah. So that's a comparison more across species and their overall size, but within the species, yes, of course, body size will have an important role, and we see it, like smaller babies will have higher frequency than their adults, yes. So this is more a comparison across species, which was a reply to Matthew Serol, because they were comparing species.
Of course. And secondly, question is, you talk about the change in terms of complexity of the whistle between species. When you talk about complexity, you are considering just, inflection points, or the secondary components, the main frequency, what you are considering when you talk about complexity?
Yeah, for that paper, the most common measure of complexity was inflection points at the time. Now we have better tools to measure complexity, and that's part of the, I think, for example, this the way to switch to the idea of vocal repertoire, for example, and use some of the tools that the bird people have been using for decades, right? Bird people have been able to get really great understanding of vocal repertoire sizes and complexity because they, they have developed all these tools. So I don't think in tooth whales we need to necessarily to invent the wheel. It just takes some of those lessons from bird people and see complexity from the point of view of vocal repertoires, which it's a big gap in our knowledge of tooth whales. It's incredible that we have 70 years of data and understand signal frequency and variation, geographical variation and behavior, but we have no idea, not even a close estimation of how big their vocal repertoires are. And I think that's so important to understand the life of these animals, like a river dolphin versus a coastal dolphin and how they relate. And I think I'm very excited, but I'm a little bit intimidated by this project, but at the same time, super excited because I think it will generate very, very novel information.
Thank you. I have more questions, but let's the other. Yeah.
Since they are not saying anything, I do have questions. That totality paper opened a can of worms, I think. And but one thing that I find interesting is that now that we get data from more and more species and we have more recording time, we are starting to understand more of their on what we thought was social or not. Like when we did with photos, we thought, everybody said that they were lonely, they were socially very simple, and they would produce very simple sounds. And then we discovered that no, they produce very modulated whistles. They can go high frequency as well. So, and on the other hand, we also have species that also were always assumed to be complex, like killer whales. And they don't use whistles at all, most of them, right? Some of them do use whistles, but most of the populations use like pulsed calls.
Yeah.
So you also have that thing
switch,
as well.
Yeah.
You switch back and forth, which is very interesting. I think that's going to be a nice challenge to think as well, because they can produce whistles, but why they don't use it? Or why do they use it in a different way? Or, what changed in the marine dolphins that
yeah, exactly. That's one thing. Because ideally we want to have a measure of the vocal repertoire that has everything, right? And I can see that in killer whales there will be whistles will not play such an important role in their communication as calls do, right? So because they probably are more represented in their vocal repertoire than whistles are. And same thing with botos, and that in itself is super exciting. So I'm trying to think about how do we incorporate that as well? Like how do we incorporate signals that not always have a specific contour? And that's a discussion that we're having with Franz Jensen, because I know his students are developing new tools that might be available. There are other tools that might also help us too. I just think that the whistles allows us to kind of get that our toes on the water, because we know so much and they're so relatively easy to extract, and then from there we can then incorporate the other sounds. But I do think you're right, there will be some points in the phylogeny where some species like phocenids, right? The phocenids have and some cephalorhynchus have completely changed the way they communicate to burst sounds, right? So what are the factors that change that? And there's hypotheses about predation and other aspects. But yeah, I think it's fascinating to think about that.
That's really, really cool. A Masa tem pergunta aí na UERJ?
Não. Ela já saiu, ela teve que sair.
E os alunos?
She's not here now.
Mas os alunos têm alguma pergunta?
Yeah. Okay. Let me see. Where is? We do not have a camera now here. I don't know why. What is going on here? But thank you very much, Laura and Beatriz. Beatriz, I'm a lecturer from this department, ecology department also, but I don't work with mammals. Sorry, and it's wonderful talking, and thank you very much. And I used to work with marine chemical ecology, and I'd like to ask about the invertebrates like barnacles, if you have some information or something like that, because I saw that some in the photos, and talk a little bit about some invertebrates. And I know that they communicate by chemical substance like pheromones and some signals and sexual pheromones, and they are a group, but however we can hear clicking and grinding sounds of the barnacles, and probably they are communicating also. And do you know about this? And do you have some information and how they talk with each other, something like that?
No, I don't know specifically about barnacle. I do know that there are many marine organisms that, as byproduct of their behavior, they produce sound like the sea urchins when they're feeding the algae, right? They produce the scrunching sounds, and some of the lobsters as well. But I have no idea about barnacles. I'll look after this because I'll be curious to find out.
Okay, thanks a lot. You are without sound, Gabriel.
I think the barnacles, they are like the sea urchins. I think it's just a byproduct of their filtering. So their shells just click. I don't think they have the brains to communicate. I have no idea. Well, I can bite my tongue.
Yeah, because they are social group about barnacles and other invertebrates. So it's okay. Thank you very much.
Thanks.
Quer falar, Ismael?
Yeah, I think let's Malu asking.
Vai, pode falar, Malu. Malu is my PhD supervisor, the other one.
Oi, hello. Congratulations for your very nice talk. And I just would like to add some information about the parrots and other social birds can do contribute with your data with dolphins. I don't know if Gabriel told you about Amazona parrot. We have more than 10 years of studies in the repertoire and social structure and the reproduction. And so I'm really glad to hear you, and I'd like to contribute with the birds data. I think just the problem is you need more birds in your talk.
Yes. So birds are not my field. I love them, but I do feel like bird people have been doing such a great job on studying vocal repertoires. I don't know specifically about parasocial structure because my understanding, and please correct me, you're an ornithologist, is that in birds their sociality is more like an individualized behavior in terms of their reproduction. But is there any acoustic behavior in parrots associated to bonding among group assemblies?
Yeah, I think so, but I'm not sure. That's very difficult to study because they used to stay also in a roosting site with, we have counted 10,000 individuals there, for example. And and take too long for the nestlings to emancipate. So they have a lot of care of the youngs and learning, and they are open-ended learner.
Yeah.
They have duets with the
male and female.
Pair, male and female. So there are a lot, and also dialects. So and also gestural communication. That's all we already published, and we have also data of other, not my work, but for example with Gira Cuckoo, we have I think large repertoires is incredible large also, and they have small groups of 12 individuals like that, and the helpers, nest helpers. So I think can contribute a little bit with the vocal repertoire and the social structure.
Yeah, it seems like it will be really interesting system to study. Vocal repertoire, composition, size with social structure. Yeah, that's really cool. Well, I'm planning to visit Brazil on my sabbatical, so I'll definitely will have to make it happen to go and see.
It'll be my honor to receive you in Belém or other places.
Ending my promotion if everything goes well. I'll make it there.
I would like to already invite you to do a symposium in the next Ethology Congress that will be online, so we can do something together, I think, because I start some studies with frequency of the sounds and the body size of many groups of birds. That information, there's a lack of information about that too. So I start with my students to work with that in many groups, and now I will start with the Piscitacid, with parrots and macaws. It'll be interesting. But I enjoy a lot of the information about the dolphins, and the data was really interesting. And sorry I arrived late, so I'd like to see the recording after, if it's possible.
Yes.
Okay.
I think parrots also produce like contact calls. It's like it's a but I don't know if it's like a bony thing or it's just for them to maintain like cohesive during the. I remember from ladies' work, right? They do produce like some specific calls when they're flying. Like part of the curve of birds as well, because they're very smart.
Incredibly smart.
They're like dolphin-like, than the other birds.
They're the dolphins of the sky, then.
Yeah.
Really, really good expression. Thank you.
Thank you.
Quer falar, Israel, a outra pergunta que tu tinha?
Okay, if anyone had more questions, I can do my third questions. I have a it's interesting when you talk about the evolutionary approach in terms of evolution of whistle also, but I saw now a bunch of papers like trying to identify the species use statistical methods. This is great. However, we have some case, actually a lot of cases, that we have exactly the same frequency counter used by different genus, for example, different species, different genus. And people are using these statistical methods even when you have, for example, mixed groups with no visual confirmation and this kinds of thing. So I really are really concerned about that, because I think their statistics is perfect, but biologically we have a problem there. So I would like to hear your opinion about.
Yeah, so you're referring that there's a lot of overlap in frequency, which is one of the parameters that we use to do algorithms, and yes, that I think that's one of the fields where this project can contribute to it. because by understanding, we're seeing that the species do vary and the composition of favor specific contours. And if we can add that to the set of variables that we're using to identify, we might get better classification. So I think that could be a byproduct of this project, because the goal is that we get, for a number of species, representation of the results across their distribution. So we'll be able to kind of capture some of that variation, kind of measure the variation, and then look at what the proportion of the common as a whole, as a species, but if that varies across different populations. And I think that will become very important to have generalized algorithms, but also locally based algorithms, right? Because they maybe that you have sympatric species of several dolphins, and then they might then diverge, and the contours that they use to be able to tell each other apart better. Like we have bottlenose dolphins and Sotalia in Costa Rica. They occupy exactly the same space. Mostly it's used by Sotalia, but when the bottlenose dolphins come from the oceanic area and they stay there, and you can tell the two species really clearly just based on the contours. Like definitely more upsweeps for Sotalia, more signs for the bottlenose dolphins. So I think you're right. I always read these papers as well, and I always wonder, beautiful statistics, right? But then, we're missing a big component, which is what are the contours, right? Which are probably more informative. Yeah.
I do agree. Alguém tem mais pergunta, pessoal? No? I think that's it. Thank you so much, Laura, for waiting for all this.
Thank you. And I really would like to extend, like, this is an ongoing project. This 5-year NSF, I have funding for field work, eventually for graduate students joining the lab, and I'm looking for students that are interested in kind of learning machine learning, taking these things to the next step, because I think it's a great opportunity also to develop computational skills that can open the job market for graduate students. I always feel uncomfortable that we are giving them as if there was just one option for future jobs, right, at the academia, which are becoming really hard. So I'm hoping that this project will bring in training in so many different areas that can open the job market opportunities for graduate students. And I think that's very important. Like, Google or whatever, just if you know how to do machine learning, AI, doesn't matter where you apply, you can get really good jobs, or apply to different things. So if there's anyone that is interested, or if you're recording species that in your area, the dolphins, that you want to contribute to some of the recordings, and we can work on collaboration agreements, so that we can work together in a kind of geographical large scale project, please let me know. Gabby knows me, and we always try to work in very ethical settings, making sure that everybody gets their recognized for their work. So if you have interest, just send me. Gabby has my email, and we can continue talking about potential collaborations.
I can make the bridge for sure.
Yeah.
Awesome. Thank you very much.
Thank you, everybody.
Thank you very much.
Thanks.
I'll stop the recording and then end the call.